is the problem with the spirit lamp. So what it is doing?
Killing microbes.
No. Spirit lamp problem is it will make you black. It will make it black as well as it's against the flow, right? Now air is flowing, so it is bending. So proper heat will not come to the tumor. And one more thing, whenever you pour, what you need to do is you need to stir it slowly, not like if you stir fastly, what happens is air bubbles will come. So the stirring mechanism in microbiology is mostly like this. Okay. So elastic here. Now it's the perfect time. Okay. It's around 50 to 60 degrees centigrade. So 60 degrees centigrade to 50 degrees centigrade is the perfect time for pouring. అంతకంటే ఎక్కువ చేస్తే ఎక్కువ మనకి హ్యూమిడిటీ ఎక్కువ అయిపోయి పైన మాయిశ్చర్ ఎక్కువ వచ్చేస్తుంది ప్లేట్ పైన. But I am not sure whether agar is in sufficient amount. But another way of pouring is there. Is like that if you pour, the problem is laminar airflow is small, and another person after you, if someone wants to work. Next one can come. ఎవరమ్మా ఫోర్ సో హ్యాండ్ ఇక్కడ ఉంది కదా ఇక్కడ హీట్ ఆన్ ఆన్